Hello, listeners. I'm going to begin by stating that I was a firm believer and researcher of evolution for a few years. I debated with several co-workers as well as friends on the subject. Upon looking into the theory, everything appeared solid and undebatable, factual even. Only an idiot would consider anything contrary. I found myself thinking that living in the Bible Belt that these people were out of touch with the modern world and reality in general. Science eluded them, and I soon found myself at odds with the beliefs of my family as well as the seniors in my community, which I expected. Once again, living in my current estate, I isolated myself and began further research in attempts to strengthen my arguments. My beliefs were agnostic, and I soon found myself questioning all the beliefs of my family and few friends. If God does exist, He made us to evolve. If not, then no worries, right? We're only animals, and seeing as how I cannot recall the previous millennia before I existed, I'm more than sure that this life is mere happenstance, and I'll simply fall into a good long sleep. So I repeat, no worries. Further research led to many interesting finds, but I soon began to notice several holes in the theory. One such unavoidable gap was the actual origin theory itself. This puzzled me. There is so very little information available. If you don't believe me, look it up. More questions began to arise in conjunction with the lack of fossil evidence provided. One inquiry was a constant. If they can discover the remains of so many dinosaurs as well as arthropods, vertebrates, echinoderms, brachiopods, why not our ancestors? I studied the likes of Lucy, Artie, Ida, the more recent Lucy's child, and began to notice a similar pattern to them as the debunked Piltdown Man, Nebraska Man, Java Man, Ernst Heckles' embryonic drawings, the horse's evolution. These findings drifted my belief further and further away from what I clinged to as a fact for so many years. My studies into DNA only strengthened the argument of a system too complex to have simply evolved. All 6 categories of evolution were personally being analyzed, and only microbacterial evolution held its ground. Macro studies seemed to hold so few facts that analysis was difficult to be placed solidly in any factual form. Fancy words are well and good for the layman people, but if my belief is lingering in the balance, I want truth and honesty. If one must disregard God for a simply more comfortable belief system, the evidence must hold firmly its own ground. I soon began looking into the possibility of intelligent design. Here are some arguments against my once thought solid belief in our origins.
The third icon of evolution that I wrote about is homology in vertebrate limbs. If you look at the bones in the human hand, the way they're constructed and put together, their structure and positions, and you compare that to a bat's wing or a porpoise's flipper or any number of other vertebrates, the structure and position of the bones are actually strikingly similar. You can sort of match them up one for one. And this was recognized before Darwin by pre-Darwinian biologists and called homology. It's different from another sort of similarity called analogy, which is that, say, between a bat's wing and a butterfly's wing. Although they're both wings and they're both used for flying, the internal structure of the bat's wing is quite different from that of a butterfly, radically different. And so analogy was not used to classify organisms, whereas homology was. When Darwin came along, he attributed homology to descent from a common ancestor, so common ancestry rather than common design. And this was at the core of his theory. He considered homology some of the best evidence for his theory, these sort of similarities in structure and position. And in fact, that's how paleontologists arrange fossils, depending on their anatomical similarities and differences. Even for the molecular comparisons, we're still talking about homology in the sense that we're comparing similarities and differences of, in this case, DNA sequences or protein sequences. So homology is at the heart of the evidence for Darwinian theory, that is, descent with modification from a common ancestor. One problem with the reliance on homology as evidence for Darwin's theory is that we have some things that appear to be homologous, but which we know or we believe do not come from a common ancestor. For example, the human eye and the eye of an octopus are actually quite similar, and yet nobody believes that the common ancestor of humans and octopuses had an eye like that. So there are cases, there's actually many cases in the biological world, of structures that appear to be homologous, but which do not come from a common ancestor. Well, Darwin's followers sort of finessed the problem by redefining homology as similarity due to a common ancestor. So instead of similarity of structure and position, homology was now similarity due to common ancestry. The problem with that move is that if you define homology in terms of common ancestry, you cannot then turn around and say that it's evidence for common ancestry, because that's circular reasoning. In effect, you're saying homology is due to common ancestry, which is due to common ancestry. So logically speaking, once you define homology in terms of common ancestry, you cannot use it as evidence for common ancestry. Although I wrote icons 10 years ago, it took many biologists a long time to realize this, but recently, Richard Dawkins, an outspoken defender of Darwinism, in his book, The Greatest Show on Earth, recognized this problem, that you cannot use a word that's defined in terms of common ancestry as evidence for common ancestry. So Dawkins' solution is to use another word, homeomorphic. So we don't talk about homology now, we talk about homeomorphism. Well, it's a nice try, but it doesn't really solve the problem. The problem is still, how do you use similarities and differences to show common ancestry as opposed to common design? As I pointed out in Icons of Evolution, the problem of using homology to infer common ancestry was illustrated by, unwittingly in this case, by Ohio State biologist Tim Berra in a book he wrote to defend Darwinism against its critics. And what Berra did was he used a series of pictures of Corvette automobiles, 54 model I think it was, 55 and so on. And he argued that by comparing the similarities in these automobiles, it's possible to know beyond a reasonable doubt, that this is descent with modification. The problem with that is that everybody knows Corvette automobiles were designed by engineers. They were not products of biological descent with modification. So what Berra unwittingly proved with his drawing is that a mere succession of similarities is not evidence for common ancestry. It could be evidence for common design. So what I wrote about homology 10 years ago in Icons of Evolution is still true. You can compare similarities and differences, anatomical, molecular, whatever. You cannot distinguish between common ancestry and common design without a mechanism. And the more we know about homology, the clearer it is that we do not have a mechanism to explain it. And so the problem that I posed in 2000 is still with us. Another famous icon that I wrote about in my book is Archaeopteryx. When Darwin published The Origin of Species, there was actually no fossil evidence for any of the major transitions that he postulated in his theory. But soon after he published the book, a now very famous fossil was discovered of a bird with feathers, beautiful feathers, that also had teeth in its beak, a long reptile-like tail, and claws on its wings. In other words, it had bird features and reptilian features, and it was widely taken to be the missing link between reptiles and birds. And it's been used that way in many biology textbooks. It's still there in some of them. We now know that Archaeopteryx has features that would have to entirely lose to become the ancestor of the modern bird. It was a side branch in the tree, if indeed there was a tree, between ancient creatures and modern birds. In fact, there's a whole school of Darwinian evolution, evolutionary biology, called cladistics, that assumes Darwinian evolution is true and then uses homology, pure and simple, to decide patterns of ancestry and descent. Forget the fossil record. We just use homology now, that is, structures and positions of features. When Darwinian biologists went looking for homologies in fossils that could plausibly indicate the ancestor of modern birds, they found little dinosaurs that had such features, but that had lived tens of millions of years after Archaeopteryx. So although Archaeopteryx was supposed to be the first bird, it turns out that its ancestors don't appear in the fossil record until tens of millions of years later. Presumably, if the Darwinists are right, these ancestors also preceded Archaeopteryx in the fossil record. But that means that the missing link is still missing. We do not have a fossil appearing at the right time in the record that could plausibly be the ancestor of Archaeopteryx and modern birds. So every now and then, as with ape-to-human fossils and various other things, we read the hype in the news about the latest feathered dinosaur found in China or something like that. In fact, here's an article that came out just this month, September 2010, about another creature found in Spain. This one has little bumps on some of its bones, and it also has certain features that are homologous to modern birds. So it's thought that this might be the ancestor that they're looking for, the missing link, the little bumps being places from which feathers sprouted, although no feathers were found with the fossil. Also, this fossil is about 20 million years younger than Archaeopteryx. So once again, we have an ancestor that's younger than its supposed descendant. And this goes on all the time. I mean, Darwinian dogma in the form of modern cladistic analysis overrides the fossil evidence and becomes evidence for Darwinian evolution. It's circular thinking all the way down. Darwinian evolution is true. This fossil fits the theory, so therefore it must be the missing link. Therefore the theory is true. And here we have it again very recently. Another icon I wrote about was a set of drawings made by 19th century Darwinist, German Darwinist Ernst Haeckel of vertebrate embryos. Now Darwin knew that the fossil evidence was not particularly helpful to his theory. So in Darwin's own opinion, the best evidence for his theory was what we see in embryos. And he believed and wrote in The Origin of Species that embryos in the same class or group are most similar in their early stages and become different only as they develop. In fact, he thought that the early stages show us the form of the ancestor in its original form. Ernst Haeckel made some drawings based on this idea that embryos are most similar in their earliest stages and become different only later on. And these drawings became quite famous. They show, typically in the first row, embryos that look almost identical. And these are embryos supposedly of fish, salamander, turtle, chicken, and various mammals. And then as they develop, these embryos become more and more different until we see the actual adult fish, salamander, turtle, and so on. Many modern biology textbooks include Haeckel's drawings as evidence for evolution. I was surprised at how many I saw. And yet, Haeckel faked the drawings. Some of his own colleagues, embryologists, accused him of fraud during his own life. The reason his drawings misrepresent the evidence, actually there are three reasons. First of all, the embryos that he portrays as being virtually identical are not. A good embryologist can see the differences, and actually in 1997, British biologist Michael Richardson and his colleagues published a now famous article in Anatomy and Embryology comparing Haeckel's drawings to actual embryos and showing that Haeckel faked the data. The other thing that's wrong with Haeckel's drawings is that he selected very carefully. He has 8 rows in his classic drawing. 4 of them are mammals. But he excluded various vertebrates that don't fit the pattern. For example, he doesn't have sharks. He doesn't have lampreys or eels, which are very different. He uses a salamander instead of a frog, because a frog doesn't look like that at all. And among the mammals, he leaves out the platypus and the kangaroo, which are different divisions of the mammal group. And he only includes mammals that happen to look very similar. So we call it cherry picking. He cherry picked the embryos he chose to show, and then he distorted them to make them look more similar than they really are. And for me as a scientist, the worst thing about Heckles' drawings is that he totally leaves out the earliest stages of embryo development. The embryos that he portrayed as being the earliest are actually midway through development. When you go back and look at the earliest stages of fishes and amphibians and birds and so on, mammals, they look very, very different. So instead of early similarity developing into later differences, we have early differences converging midway on certain similarities, which he exaggerated, and then diverging again as the embryos develop. It doesn't fit Darwin's pattern at all. In fact, if you were to follow Darwin's logic, you'd have to say the different classes of vertebrates had different ancestors, and then for some unknown reason become similar halfway through development and then become dissimilar again. So the logic and the evidence don't fit. So after I wrote about Heckles' embryos in 2000, the pattern among people who were critical of my book became quite typical. First, they started quietly taking the drawings out of the textbooks. Okay, Heckles' embryos are hard to find in textbooks now, although there's actually still some that have them. Or they replaced Heckles' drawings with redrawn versions, or in some cases with actual photographs, but the photographs are only of those embryos and those stages that happen to fit the Darwinian story. So the evidence is still being widely misrepresented in textbooks. At the same time, I was personally vilified by many of my critics as a liar, as being stupid. I'm actually trained as an embryologist, and yet they said, obviously, I didn't learn anything from my Berkeley PhD because I didn't know anything about embryology. Interestingly enough, Jerry Coyne, who is a fly geneticist, when he attacked my book, he attacked me primarily, as far as the science goes, on my portrayal of vertebrate embryos, my own specialty. But perhaps the most egregious attack on me and my credibility came with a film called Flock of Dodos by evolutionary biologist turned filmmaker Randy Olson. And in the film, he has me on camera as saying that the embryos misrepresent the truth and the textbooks still have them. And then he tries to show in subsequent scenes that the embryos really are not in the textbooks and that no biologist takes them seriously anymore. Well, he knew at the time, because Icons of Evolution actually cites 8 textbooks with Heckles' embryos, he knew that what he was saying wasn't true. And he was later then confronted with other textbooks, more recent ones, that still have Heckles' embryos in them. And he decided to go ahead with the film anyway because he liked the story he was telling. But it has absolutely nothing to do with the truth. The truth is, Heckles' embryos were used in textbooks all over the place for a century after they were known to be fake. And I think this is an outrageous example of how some Darwinists misrepresent the evidence to prove their theory. One of the icons of evolution that I wrote about is the 4-winged fruit fly. Now, fruit flies normally have 2 wings, and behind those wings are a pair of what are called balancers that vibrate, oscillate rapidly in flight to stabilize the fly's flight. But if you combine 3 mutations very carefully in a fruit fly embryo, you can produce a fruit fly with 4 normal-looking wings, 2 pairs. Ed Lewis did this and won the Nobel Prize for it, deservedly, because it showed us a lot about the developmental genetics of fruit flies. In terms of evolution, however, it shows us nothing, because the second pair of wings has no muscles. It's dead. It's like having a small plane with an extra pair of wings hanging off the tail, trying to take off from a runway. So the four-winged fruit fly is actually a cripple. And it turns out, this is why the icon is important, even though it's not as common in textbooks as the other icons. What's interesting is we have many cases of mutations that produce minor biochemical changes, such as antibiotic resistance in bacteria. But we do not know of a single anatomical mutation that's beneficial to the organism. People have mutated fruit flies every way they can. They've done the same with zebrafish. They've done the same with a roundworm called C. elegans. They've done the same almost completely now with mice. It's called saturation mutagenesis. There's only three possible outcomes, we now know. If you mutate an organism and watch it develop from an embryo, there's only three possible outcomes. If you mutate a fruit fly, you either get a normal fruit fly, a defective fruit fly, or a dead fruit fly. That's it. Those are the only three possibilities. You don't create new organs or body plans. You don't even change the species. So mutations are highly overrated as a source of raw materials for evolution, and that's the point of my fruit fly chapter. The second icon of evolution I wrote about is Darwin's Tree of Life, which is actually the heart of his theory. The only illustration he put in the Origin of Species was this branching tree pattern that supposedly shows how all living things descended with modification from a common ancestor at the root. Some of the evidence Darwin cited for his Tree of Life was the fossil record. Supposedly, if his theory were correct, if we look at the fossil record, we would see, first of all, a common ancestor and then these multiple branches coming off with, as Darwin put it himself, innumerable transitional steps in between. Because in Darwin's theory, a species gives birth to, or an organism gives birth to another organism very much like it, and only over many thousands or millions of generations do the differences appear that then result in new species and new classes and orders and so on. Well, Darwin himself knew that the fossil record did not fit that picture. He knew, for example, about the relatively sudden appearance of the major forms of animals in the Cambrian, a geological period known as the Cambrian. We now call those major forms phyla. For example, we are in one phylum, that is a phylum that includes animals with internal skeletons and soft bodies. Another phylum would be the crustaceans, or the insects, which have an external skeleton and a soft internal body. There are the starfish, there's various worm phyla, there's the mollusks that include clams and octopuses. And all of these phyla have fundamentally different body plans. In Darwin's theory, the first original ancestor would have been one of these, perhaps, one phylum. And the big differences that we now see in the different phyla would have emerged only after millions of generations. And yet, what we see in the fossil record is the major animal phyla appear at about the same time in the Cambrian Explosion, what's now called the Cambrian Explosion. Darwin thought the problem could be explained away by the incompleteness of the fossil record. Earlier forms had existed, they had to exist according to his theory, but they were too soft or too small to be fossilized, and so we cannot find them. But since Darwin's time, fossil hunters have found lots of fossils below the Cambrian. They're very small, soft-bodied. Some of them are bacteria that go back billions of years. So the idea that the fossils below the Cambrian, earlier than the Cambrian, have disappeared is simply false. We have found them. So the fossil record now shows that up to, from about the origin of life, up to just before the Cambrian, we have single-celled organisms, very small. And then in a relatively short period of time, we get the major animal forms. I'm not talking about all the animals. Mammals weren't there, for example, but vertebrates were there, animals with backbones, right there in the Cambrian Explosion. The suddenness can be illustrated perhaps by thinking of a football field. If you were to start at one end of a football field and walk toward the other, and the football field represents the entire history of life, you'd walk 25 yards, the first line is the origin of life. The first 25 yards, small single-celled organisms. The next 25, same thing. The next 25, same thing. You've gone 75 yards, and all you have are small single-celled organisms. And then somewhere around, I think it's the 84-yard line, in the space of a single stride, the major animal forms appear. And those major forms, for the most part, are with us today. This is not a branching tree pattern, as Darwin's theory demands. This is more like a lawn than a tree. Darwin hoped that with future fossil hunting, either the transitional forms that he postulated would be found, or it would become absolutely clear that the fossil record was destroyed before the Cambrian. And both of these have turned out to be false. The transitional forms are not there, and the fossil record before the Cambrian is not all that bad. So in terms of fossils, Darwin's theory does not fit the evidence. The first icon of evolution I wrote about was the Miller-Urey experiment from 1953. Darwin himself actually didn't talk much about the origin of life. His theory began with the first living cell and went from there on. But he hinted, Darwin that is, hinted at the origin of life in some, what he called, warm little pond. In the 1930s, various people came up with an idea that life began on the early Earth when lightning created organic molecules in the atmosphere, which then dissolved in the ocean to form what they called a primordial soup. Well, this was all hypothetical until Stanley Miller in 1953 performed an experiment in which he put in a glass apparatus a mixture of gases that he and others thought represented the early Earth's atmosphere. And then he shot an electric spark through that and collected the products in water at the bottom of the apparatus. And after about a week, he was able to detect two of the simplest amino acids, which are the building blocks of proteins. When I wrote Icons of Evolution in 2000, many biology textbooks had images of the Miller-Urey experiment, because although Darwinian evolution doesn't start till after the origin of life, there was this grand evolutionary story that the textbooks wanted to tell, and the Miller-Urey experiment was part of that story. There are many problems with the Miller-Urey experiment. He used a mixture of gases, methane, ammonia, water vapor, and hydrogen in his apparatus that many scientists at the time thought represented the early Earth's atmosphere. But by a decade later, most geoscientists, or many geoscientists, were convinced that the early atmosphere was not like that at all. First of all, it seems there was probably some oxygen present. Well, if you had put oxygen in the Miller-Urey apparatus with hydrogen and then shot a spark through it, the thing would have blown up. So oxygen had to be excluded. And yet it appears now that probably there was some oxygen in the early atmosphere. The early atmosphere apparently was not rich in hydrogen. Most geoscientists now think the early atmosphere consisted of the same gases that come out of volcanoes in the modern world. And those gases are water vapor, carbon dioxide, and nitrogen. Well, if you put those gases into the Miller-Urey apparatus and shoot a spark through them, you don't get any amino acids at all. You have to have some free hydrogen present. So when you put a realistic mixture of gases in the Miller-Urey apparatus, you don't get anything like the building blocks of life. And so the experiment appears now to be irrelevant to the origin of life. But there's an even more serious problem. Even if someone could show, and every now and then someone publishes a paper claiming to show, that certain basic building blocks of life could form under pre-life conditions without any sort of intelligent design or intervention. Even if we could show that, we would still be immeasurably far from creating a living cell. And here's how we know that. And this was published even before Icons of Evolution. If you take a little flask, a little test tube, and you put in a sterile buffer that's a salt solution with no living thing in it at all, then you add one living cell to that sterile solution, and you poke a hole with a sterile needle. The contents of the cell will leak out into the buffer, and you have in your hand all the molecules you need for a living cell, because the cell was just alive a minute ago. Not just the building blocks, the DNA, the proteins, the membranes, everything is there. And yet, every scientist knows you cannot put that back together and make a living cell out of it. I call it the Humpty Dumpty experiment. So even if we could show how all the chemical building blocks of life formed on the early Earth without any intelligence or design, we would still be immeasurably far from understanding how life began. Because we can have all the chemicals present and still not know how to make a living cell. The whole thing remains a mystery. The last icon of evolution that I wrote about, I called the ultimate icon. It's the famous ape-to-human progression. And the reason I call it the ultimate icon is because it's the one that really matters to someone who wants to tell a Darwinian tale, that is, a tale about how we came here accidentally as a byproduct of material causes. So typically the icon shows a hunched-over chimpanzee-like creature gradually morphing into an ape man and then a modern human. There's many forms of this icon. You can find it everywhere, including many textbooks. But the evidence for the ape-to-human icon is surprisingly thin. We have a great fossil record of shellfish, of various other kinds of animals, but we do not have a great fossil record of the creatures that supposedly provided the transitions between fossil apes or ape-like creatures and modern humans. There are some fossils, yes, but what happens really, and experts in the field of human paleoanthropology have said as much, we start with a story. The story is that apes or ape-like creatures evolved into humans. We find things to plug in there. For example, before Darwin had any evidence at all of any of this, his friend and defender, Thomas Huxley, used a series of skeletons of apes that supposedly showed a progression up to the human skeleton. Well, these are all modern apes. They're not ancestors and descendants, and he didn't claim they were. But just this mere comparison of similarities, which I pointed out earlier in the chapter on homology, does not really distinguish between common ancestry and common design. This pattern of morphing into the human skeleton then had to be filled in over the years with various fossils that were turned up mostly in Africa. What's funny is that many of these fossils can be interpreted in various ways, to look various ways. Often a fossil which is initially proclaimed to be, the key to the whole story is either fake, in the case of Piltdown Man, which was a complete fraud, or the fossil turns out to be a part of a different branch of the primate evolutionary tree. In 2009, a fossil was widely advertised as being, an important link in the ancestry of humans. The scientific name was Darwinius massilae, and it was nicknamed Ida, and got a lot of press. It actually looks like a small cat. It's a lemur, which is a primate, although not an ape. And it turns out that they started the promotional campaign 6 months or so before announcing the fossil in the scientific literature. So they had this big promo campaign planned for Ida. They put out the evidence, and almost immediately, evolutionary biologists began attacking it. They said, this is nonsense. This is not part of a human evolutionary tree. This might be important in deciding one minor issue in primate evolution, having to do with the classification of lemurs. But it's not an ancestor of human beings. It had no descendants, for one thing. And so the fossil was quickly discarded. And almost immediately, they had another one to take its place called Ardipithecus ramidus, which is a much more plausible one to stick into the story, but which has also been known for 15 years. Why did it all of a sudden become, the subject of all this hype? Obviously, there's much more to the ape-to-human icon than just scientific evidence. In all these cases, we have a story that's in need of evidence. And so whatever fossil turns up that appears to fit that story is hyped and milked for as much as Darwinists can get out of it. And then maybe something else turns up and gets the news after that. The thing that bothers me most about the idea of an ape-like creature evolving into a human is we have no mechanism, no plausible mechanism to explain that. The evidence we have for natural selection shows only changes within existing species. The evidence we have for mutations doesn't get us new organs or body plans. So the idea that this evolution could happen, to me, rests mostly on the claim that there's a mechanism, and we don't have that. Barring that, it's just a story into which we insert various pictorial illustrations. And that's what the ape-to-human icon is about. I wrote about 10 icons of evolution in my book. I could have written about more, but I had to stop somewhere. And the 10 that I wrote about were, first of all, the 1953 Miller-Urey experiment that purported to show that life could have begun spontaneously on the early Earth. And this is still found in most biology textbooks, even though geochemists decades ago decided that the experiment was unrealistic and really doesn't say anything substantial about the origin of life. The second icon was Darwin's tree of life, the branching pattern that would show how all organisms descended from a common ancestor at the root. And this is a theoretical statement, but it's presented in most biology textbooks as though it were a fact. The fossil evidence, for example, does not show this branching tree pattern. When you look at the different major groups of animals, they appear pretty much at the same time, rather than diverging from a common ancestor. The third icon I wrote about was homology in vertebrate limbs. If you look at the bone structure of the human hand or the human arm, you compare that to a whale's flipper or a bird's wing or a bat's wing, there are certain striking similarities in the bone structure. And Darwin considered this to be a result of common descent. But people before Darwin had noticed the same thing, and they called it, they attributed it to a common designer. Well, the truth is, it could be either one. The bone structure itself doesn't tell us. The fourth icon is Heckles embryo drawings, which purport to show that vertebrates are very similar as early embryos, and therefore this provides evidence for common ancestry. But in fact, the embryos are not similar, and Haeckel faked the drawings. The fifth icon that I wrote about was Archaeopteryx, a famous fossil of an ancient bird that had a long tail and teeth in its beak like reptiles, and for a long time was thought to be the missing link between reptiles and birds. Biologists no longer think that because there are too many differences between Archaeopteryx and modern birds, but textbooks tend to present it as the missing link anyway. The peppered moth is a famous icon of evolution in which moths supposedly changed color during the Industrial Revolution because dark moths were better camouflaged on soot-darkened tree trunks, and the birds couldn't see them, and so they ate the light moths and left the dark ones. And this is still used in many textbooks as a classic example of natural selection in action, when in fact biologists discovered in the 1980s that peppered moths don't rest on tree trunks in the wild at all, or normally. And so the textbook pictures have all been staged, and the story has serious flaws. Darwin's finches, another icon of evolution, are some species of birds on the Galapagos Islands that are very similar except for the size and shape of their beaks. And it's thought, and it's quite possible, that they evolved from a common ancestor because of having to eat different foods on the different islands. But the actual evidence shows us only that the beaks can change over a matter of years based on climate and diet. But the changes are temporary, they oscillate back and forth, and they don't go anywhere. So as evidence for the origin of species, Darwin's finches really don't work. Another icon of evolution is the four-winged fruit fly. Most fruit flies have two wings, but a series of mutations in a fruit fly can cause it to develop a second set of wings, very normal looking, but in fact there are no muscles attached to it, so the second set of wings is effectively dead. The fly is a hopeless cripple. It's kind of like having a small plane with an extra pair of wings tied to its tail. This is not the forerunner of a new race of insects, but an evolutionary dead end. The last two icons I wrote about were fossil horses and the ape-to-human picture. In both cases, there is some real evidence here. The important point in both of these icons, I'll mention them together, is that a very heavy dose of philosophy is laid onto the evidence in each case, mainly to persuade students that evolution is without direction or purpose. In the case of the ape-to-human icon, students are also told that humans, in effect, are nothing more than animals. Neither one of those claims comes from the evidence. Both of those are philosophical claims, but they use the evidence to make themselves appear more scientific than they really are. And so that's my criticism of those two icons. I think the most important reason to question and criticize Darwinian evolution is to make science better. The issue here is how to do good science. By that I mean theory, evidence, checking one against the other, and being willing to abandon the theory when it doesn't fit the evidence. That to me is science. It's ultimately truth-seeking. Darwinism, to me, is ideology, indoctrination. It's theory at all costs.
There is usually an element of the truth in any false teaching, which is used to try to convince us that the whole teaching is true. Therefore, we need to think clearly and pay attention to be certain that conclusions are really supported by the evidence. The argument is not over the evidence, but their interpretation. The icons may be interesting, but they really have nothing to say about the real origins of man. The icons' continued and repeated use demonstrates the lack of real scientific evidence supporting evolution. As science has advanced to demonstrate that all living organisms are based on complex information systems requiring an intelligent agent, it's tragic so much time and money is being wasted trying to support an atheistic worldview based on an antiquated 1859 theory. Who is really stuck in the past? Then we will no longer be like children, forever changing our minds about what we believe because someone has told us something different. or because someone has cleverly lied to us and made the lie sound like the truth. Ephesians 4:14.
Well, then who did create the heavens and the earth?
Why do you use the word who? You see, you immediately beg the question by using the word who.
Well, then how did it get created?
Well, by a very slow process.
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
And what was that?
It was the origin of the first self-replicating molecule.
Right, and how did that happen?
I told you we don't know.
So you have no idea how it started?
No, nor has anybody.
Nor has anyone else. What do you think is the possibility that intelligent design might turn out to be the answer to some issues in genetics or in evolution?
It could come about in the following way. It could be that at some earlier time, somewhere in the universe, a civilization evolved by probably some kind of Darwinian means to a very, very high level of technology and designed a form of life that they seeded onto perhaps this planet. Now, that is a possibility and an intriguing possibility. And I suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of designer.
Wait a second. Richard Dawkins thought intelligent design might be a legitimate pursuit?
And that designer could well be a higher intelligence from elsewhere in the universe. Well, but that higher intelligence would itself have had to have come about by some explicable or ultimately explicable process. It couldn't have just jumped into existence spontaneously. That's the point.
So Professor Dawkins was not against intelligent design, just certain types of designers, such as God. So the Hebrew God, the God of the Old Testament, he doesn't exist in your view?
Certainly. I mean, that would be a very unpleasant prospect.
And the Holy Trinity of the New Testament,
nothing like that.
And the foremost atheist in the world now believes science is providing evidence for the very existence of God. Many scientists and atheists believe science proves there is no God. Those who believe in God's existence say he created the universe and life itself. This battle between two worldviews has raged in the fields of our classrooms across the country between creationists and Darwinians for decades. At a recent summit at New York University, Flew changed his position and now believes in God as the creator of the universe.
It is all a matter of the enormous complexity by which the results are achieved, which look to me like the work of intelligence.
Flew discussed the points with Professor Gerald Schroeder, who was with the Weizmann Institute of Jerusalem and MIT, and Professor John Haldane of the University of St. Andrews. Flew turns to various discoveries of science to prove his point. From the fossil records showing the sudden appearance of a fully developed life to the emergence of visual consciousness across the animal kingdom to the basic need for reproduction. But it is the manifestation of life written in DNA and the transcription of DNA to RNA and RNA into protein and the subsequent process of protein folding that makes the best case for Flew.
What I think that the DNA material has done has shown by its almost unbelievable complexity of the arrangements which lead to produce this being, that intelligence must have been involved in getting these extraordinarily diverse elements to work together.
By requoting Flew,
which looked to me like the work of intelligence.
The Cambrian Explosion has generated extensive scientific debate. The seemingly rapid appearance of fossils in the primordial strata was noted as early as 1840s, and in 1859 Darwin discussed it as one of the main objections that could be made against this theory of evolution by natural selection. The long-running puzzlement about the appearance of the Cambrian fauna seemingly abruptly and from nowhere centers on three key points. Number 1. Whether there really were a mass diversification of complex organisms over a relatively short period of time during the Cambrian. 2. 2. What might have caused such rapid changes? 3. What it would imply about the origin and evolution of animals. Scientists cite interpretation as being difficult due to a limited supply of evidence based mainly on an incomplete fossil record and chemical signatures remaining in the Cambrian rocks. Now let's take a look at the second law of thermodynamics. Albert Einstein called it the only physical theory of universal content which I am convinced will never be overthrown. End quote. Seth Lloyd stated that nothing in life is certain except death, taxes, and the second law of thermodynamics. So it's solid. The law states that anything left into its own tends to get worse and not better. If you leave a child in a room with toys in a box for 3 minutes in return, the toys aren't going to be in the box anymore, and the room's going to look a little bit worse. Right, moms? So how can we expect to be alive on such a beautiful and wonderful planet, thriving with life, waking up day after day, night after night, peddling with all our small little problems and insignificant issues, all the while we're racing at 66,000 miles per hour around a flaming star, and we still remain thinking that no one's on watch? Come on, people. Really? I'd now like to quickly inquire as to a couple of examples given by Ken Hovens and others on the matter of how the termite and woodpecker came about evolving. The termite, as everyone knows, enjoys feeding on wood, as well as leaf litter, soil, and animal dung, but what most do not know is they cannot digest the wood. Therefore, tiny little critters, Hovens' term, called flagellate protists must eat the wood inside of the termite's gut for cellulose digestion. Knowledge on the relationship between microbial and termite parts of their digestion is still rudimentary in the scientific field of their study. The question is, which one evolved first? Without the critters, the termites would die, and without the termites, these critters wouldn't exist. Now for the question of the woodpeckers. The evolutionary history of woodpeckers is lacking in many categories, but the tongue of the woodpeckers is most intriguing. Their tongue begins in their nostril, wraps around the top of their head, over the back of their neck, under the base of the jaw, and out of the beak. The question is not if they evolved, but what it evolved from. Surely it wasn't born perfect. According to evolutionists, everything had to evolve, right? One thing that evolution has impacted most is our perceptions. Let's take a moment to compare a pin and a strand of grass. If one were to ask you which one were intelligently designed, the obvious answer to the modern world would be pin. The plastic, rubber, ink, and steel, etc. have all been formed together in order to make a convenient tool used for writing, so the complexities in conjunction with its purpose defines it as intelligently designed. Okay? Now, let's examine a grass strand. Notice the epidermis, vein, midvein, guard cells, its bundle sheath, etc. A single strand of grass is far more complex than any pin ever constructed or ever will be constructed. It is the most widespread plant type. It's the dominant vegetation in most habitats. It's the main source of food for many types of animals. Its agricultural importance has fed domesticated animals, according to Wikipedia.com, for up to 6,000 years. It has also been used for paper making since 2400 BC or before. It can adapt to conditions that the pin would stand no chance, such as in lush rainforests, dry deserts, cold mountains, and even intertidal habitats. The list goes on and on. So I ask you again, which one is intelligently designed?
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rhodobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell surface, make up the three parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains two mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the mote B proteins. This binding causes a conformational change in mote A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fle-F come together in the plasma membrane to form the MS ring. The Fle-G proteins assemble under the MS ring. Fle-G, along with Fle-M and Fle-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mote A and mote B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but mote B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
Sight, a precious gift we can't take for granted. Our eyes are windows to the wonders of the world. All we know and love, experience and discover, ponder and cherish. Let's peer into the workings of the eye, a unique and self-sustaining system. The reflected light of the world enters through the crystal transparency of the cornea, aqueous humor, lens, and vitreous humor to project onto the photoreceptors of the retina, whose impulses converge on the optic nerve, and then to the brain, to be transfigured into imagery and imbued with meaning. Continual adjustments to the pupil and lens regulate the entry and focusing of light. In today's world, the eye's surface is constantly challenged to protect itself and adjust to changing conditions. Every blink helps our eye's natural defense system, the tear film, retain moisture and maintain visual acuity. Tears contain potent natural disinfectants to keep bacteria at bay. And the tear system washes away impurities to maintain eye health. The miraculous biology of our eyes inspires us to develop ways we can protect and maintain comfort for these remarkable structures. For all the wonders our eyes provide throughout our lives, we owe them our attention and ongoing care.
Let's go further into the eye. Darwin admitted himself that the eye seems so advanced that it would not have formed by natural selection. We know a great deal more today than he did. One study of modern ophthalmology is that there are 3 almost imperceptible tiny eye movements: tremors, drifts, and saccades, and these are caused by minute contractions in the 6 muscles attached to the outside of each of our eyes. Every fraction of a second, they very lightly shift the position of your eyeball automatically without conscious effort on your part, making sight possible. The tiniest and most intriguing of these movements are tremors. They continuously and rapidly wobble your eyeball about its center in a circular fashion. They cause the cornea and retina, the front and back, of our eyes to move in circles with incredibly minute diameters of approximately 1 in 1,000 of a millimeter. This size is about 70 times smaller than the thickness of a piece of paper. Another characteristic of tremors is that the seemingly tireless muscles that produce them wobble your eye 30 to 70 times each second. On average, each of our eyes complete 1 million of these tiny circular motions in 5.5 hours. Even though they can only be seen through magnification, we could not see without them. Consider if all these movements suddenly stopped. While staring at someone's face, the light-sensing cells in your retina would quickly stabilize and cease to send updated information to your brain, causing the image you perceive to fade into a uniform gray within seconds. If the person smiled, their mouth and only their mouth would momentarily reappear out of a visual field of nothingness. The reappearance of only part of their face would happen because only the mouth moved, causing a momentary change in that part of the picture which the retina was seeing at the time. Thus, the continued change in the light projected on each retinal cell in our eyes is crucial for constant vision. Hence, the need for the tremors to supply the retina with a slightly shifting picture many times each second. During drift movements, the eye drifts relatively slowly and smoothly off the target where you are looking until it reaches an angle equal to about 12 times the size of a tremor. At this time, the eye automatically jerks via a saccade back to its original position. Saccades, which happen up to several times a second, are very quick, jerk-type movements that are used to correct for whatever drifts are occurring. We can observe the effect that drifts along with associated saccades have on our visual system by studying the graph here, or others like it. Our eyes are moving constantly, even when we think they are not. Look intently at this graph's center. You should see a slight, shimmering psychedelic effect. This seems to jump about no matter how hard you work at holding your eyes steady. Large saccades are employed in scanning motions like reading. As you read, you think that it's moving, scanning, letter by letter and word by word, but it is not. Instead, the precise alignment of your two eyes is synchronously hopping along via the jerkback saccades, following each line. During the movement, a saccade is occurring. Your vision is blurred, so between the hops are momentary stops, which give the eye-brain system time to decipher the printed letters into meaningful phrases. Just think of how challenging it would be for a human to create the genetic code needed to produce the fine-tuned nervous system that makes precise coordinated muscular movements like tremors, drifts, and saccades possible. Many evolutionists have tried to say that the eye evolved from back to front. I'll now read for you a quote from Dr. George Marshall, an eye disease researcher from the University of Glasgow, Scotland. The light-detecting structures within the photoreceptor cells are located in the stack of discs. These discs are being continually replaced by the formation of new ones at the cell body end of the stack, thereby pushing older discs down the stack. Those discs at the other end of the stack are swallowed by a single layer of retinal pigment epithelial cells, or RPE. RPE cells are highly active, and for this they need a very large blood supply. This comes from the choroid. Unlike the retina, which is virtually transparent, the choroid is virtually opaque because of the vast numbers of red blood cells within it. For the retina to be wired the way that Professor Richard Dawkins suggests would require the choroid to come between the photoreceptor cells and the light. The RPE cells must be kept in intimate contact with both the choroid and photoreceptor to perform their job. Anybody who has had the misfortune of hemorrhage in front of the retina will testify as to how well red blood cells block out the eye. The idea that the eye is wired backward comes from a lack of knowledge of eye function and anatomy. The more I study the human eye, the harder it is to believe that it evolved. Most people see the miracle of sight. I see the miracle of complexity on viewing things at 100,000 times magnification. It is the perfection of this complexity that causes me to balk at evolutionary theory. The retina is probably the most complicated tissue in the whole body. Millions of nerve cells interconnect in a fantastic number of ways to form a miniature brain. Much of what the photoreceptors see is interpreted and processed by the retina long before it enters the brain. When Darwin made his assumptions about the origin of organs, he had nothing like the knowledge that we have today. Had he been aware of the need for the tiny precision, hopping motions that are going on all the time while we are awake, he may have abandoned his theory as impractical speculation.
When an abundant supply of oxygen is available to a cell, fermentation does not go on at all, but rather cellular respiration occurs. Cellular respiration occurs not in the cytosol of the cell, but rather in organelles called mitochondria, which in some ways can be compared to the chloroplasts of plants. But rather than building glucose molecules, they break glucose molecules down into carbon dioxide and water. Mitochondria have two membranes, producing two compartments. An inner compartment, enclosed by the inner membrane and containing the mitochondrial matrix, and an intermembrane compartment between the inner and outer membranes. The first step of cellular respiration is glycolysis, which we looked at earlier. As you'll remember, glycolysis takes place in a cell's cytosol and produces 2 molecules of pyruvic acid per molecule of glucose. In order to proceed with the process of cellular respiration, the 2 pyruvic acid molecules produced by the division of each glucose molecule must be transported through both mitochondrial membranes into the mitochondrial matrix. Large pores in the outer membrane of the mitochondria make it highly permeable to pyruvic acid molecules. Pyruvic acid molecules cross the inner membrane through special proteins. Once within the matrix, each of the 2 pyruvic acid molecules is split into a CO2 molecule and a 2-carbon molecule called an acetyl group. Each acetyl group immediately attaches to a molecule called coenzyme A, forming an acetyl coenzyme A complex, or acetyl-CoA for short. During these reactions, an energetic electron and a hydrogen are transferred to positively charged NAD+ ions, forming one energy-carrying NADH molecule per acetyl-CoA. The 2 acetyl-CoA molecules enter a cyclic pathway, known variously as the Krebs cycle, referring to its discoverer Hans Krebs, or the citric acid cycle, named for the first product in the reaction sequence, citric acid. We'll refer to it as the Krebs cycle.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrath of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
So, in conclusion, I encourage those of you whom seek truth to gather together your own information through your own research, because my personal opinions are just that, my personal opinions. The purpose of this video is simply to provide information on this theory. Much more lingers in the balance here than you think. You're not a primordial soup, you're not a chimp. You are created, you are maintained, and on your very last day on this earth you will face the Creator. Be prepared, for the part of you that is to die shall die, but the part of you that won't will not, and you'll face Him. Thank you for watching and listening. God bless.